Hello again, people. So this is the video about taxonomy of microorganisms. And part of why we look at that topic is because it includes how we write the names of microorganisms and how we say the names of microorganisms. And taxonomy is the field of research where people classify organisms and name them. And so we need to understand there is a good reason for that. There's a reason we need to classify and agree on their names. And so part of it is we have to say them correctly and write them correctly. And part of it is we all have to agree on what we're talking about. So we start just by looking at how we write their names and know that this is important as any medical term you would learn before going into work in a clinic. You have to use them right or people will look at you and wonder, why don't you know what you're talking about? Just little things like plural versus singular end up being very important in people's opinion of how much you know. So got to be careful about these things. So how we write species names? Well, there are two parts to a species name. One of them is the genus. So in Escherichia coli, Escherichia is actually the genus, and there are other species within that genus. And then the second part is the specific epithet, or coli. And that isn't, the specific epithet is just a name, but there's no group coli. Coli isn't anything. It just modifies Escherichia to show you which species it is. And when you write them, notice that you will italicize or underline both the genus name and the specific epithet. When I type them out, I always italicize them because it's easy, but when I write them, I usually underline them. And the genus is capitalized and the specific epithet is never capitalized. So it's funny when I see these things written out, someone will write about this, but they won't know how to format it. That's a clear way of knowing, well, that person doesn't have any formal training in this field. They've never taken a microbiology class or anything like that if they capitalize coli. So do it right. Do it right. So you can shorten Escherichia coli to E. coli if people reading it would know what that E stands for. You presumably would have already written out Escherichia. On a quiz or exam in this course, you would never abbreviate anything. But it is permissible when you write about a microorganism to abbreviate the genus once you have spelled it out once. And so, yeah, if you're naming a species, you write the genus and the specific epithet. If you just write the genus, you're just talking about the genus. And everything you say about this would have to be true about all the species within that genus. If you just write the specific epithet, it's meaningless and you're making a mistake. So you have to write Escherichia coli if you're talking about that species, or E. coli. If you write Escherichia, it's the genus. And if you just write the specific epithet coli, you're not saying anything. So be careful about these things. So genus is a thing we're going to see many times. We will talk about many different genera. Genera is the plural form of genus. And so we will look at several different genera. Big ones we will think about are Clostridium, Bacillus. In both of those cases, there will be a lot of different species we think about. And all of them have the same characteristics we will care about. Also, there are several different species of Streptococcus that we'll look at. And those are probably the big ones. Clostridium, Bacillus, and Streptococcus are the different genera we will care about the most. Okay, so that's a good way to start this. Now we'll look at taxonomy itself, not just naming, but taxonomy. It is the classification and organization of organisms into groups. So the example I give here is like if you study whales and you're a whale researcher and you're going to go write an article about something you learned about whales, it would be really dumb if you wrote, I discovered a new fact about a shark. You know, if you suddenly forgot the difference between whales and sharks and you put shark in the title of your article, the wrong people would read it. It wouldn't make any sense, right? This is a silly example, but you have to agree with the people you're talking to about what you're talking about. So you have to use terms that they understand and that you all agree mean what you want them to mean. And so other examples would be the names of viruses like SARS-CoV-2, which causes COVID-19. That virus has a similar name to SARS-CoV, which causes the disease SARS. And so we have to be very careful when we write about those things. So when you are treating an infectious disease, there are times when if you can figure out what species is causing the disease, you can figure out how to treat it and how to respond. In many cases, you have to start treatment before you know, but there are some cases where it could make a big difference. So one example is Yersinia pestis. If you find out that Yersinia pestis is what is making your patient sick, you'll know you have to isolate them because they have plague. Things like that. Taxonomy is important for figuring out which antibiotics to use. In a lot of cases, clinicians will have good guesses about what is causing a certain type of infection, and they'll have good guesses about which antibiotics are likely to work. But if you want to narrow it down to narrow spectrum antibiotics, it can help to know the species name. Okay, so we're going to look at a lot of different groups in this course. We're going to look a lot at bacteria and a little bit at archaea. We will look at protists and fungi in one of our lectures, and we'll also look at helminths. And then in another lecture, we'll look at viruses and prions. And then when we think about infectious diseases, we will look at a lot of them. So these are different groups, but they all have kind of interesting taxonomy. So bacteria is a domain. Archaea is a domain. Fungi is a kingdom. So what does that mean? Domain, kingdom. And protists is nothing. It's not actually a taxonomic group. So that's kind of fun. So again, we have domains, kingdoms, phyla, lots of different taxonomic levels. And we start with the biggest levels. Those are the domains, and there are 3 domains. Eukarya, that's the one for all the species whose cells have nuclei, like us. And fungi, algae, protists, these are all potentially single-celled organisms in eukarya. But there are also single-celled organisms that don't have nuclei, that don't have any internal membrane-bound organelles. So the prokaryotes, and those are the bacteria and the archaea. But those are separate domains. These are very distantly related from each other. Even though under the microscope they might look the same, they're not related to each other very recently. So it's important, review eukaryotes and prokaryotes, go back to your notes and compare what is different between them. And let's look at the taxonomic levels. So let's look here. Taxonomic levels are this hierarchy where we can say within, for example, within a domain there are many species. Within a genus there are several species. Within each species there might be strains, and these are the levels in order. Domain is the biggest level. Under that there are kingdoms. We don't use kingdoms in bacteria, which is what we think about most often, but we do use phylum. This is the singular. Phyla is the plural with an A. Phyla would be plural. And then within each phylum, there are classes, and within each class, there are orders. Within each order, there are families. In each family, there are genera, and within each genus, there are species, and within each species, there are strains. And so the two different parentheses I put here, kingdom is because we don't use them in much in microbiology, and strain because you might not use this outside of microbiology, but we do use it very often in microbiology. So part of what you're having to do is think about the differences between strains. So really the taxonomic levels we're going to care about are going to be species, genus. So we're going to care about genus. I already mentioned Clostridium, Bacillus, Streptococcus. Species we're going to care about, like the difference between Streptococcus pyogenes and Streptococcus pneumoniae. And then strains. Strains are really important in E. coli. Within that species, E. coli, there are strains that people eat as probiotics, and there are other strains that if you ate them, they would kill you. It's just amazing, the diversity of strains within that one species, E. coli. So, that's cool. So, genus, again, the plural of that is genera, and these are some examples. Strains are the smallest group. Well, let's just go back, sorry. For genus, the way you write them again is you capitalize and italicize. So you capitalize the first letter and italicize. And you'll notice if we, again, with species, we capitalize and italicize the genus name and then italicize the specific epithet, but we don't capitalize that. Then strains are the smallest groups, and we don't we don't have any rule for how we format strain names. So these are example strain names: Nissel 1917, 0157H7, and K12. Those are all known strains of E. coli. And so you would write them as Escherichia coli, and then you'd write one of these strain names. And then there are larger groups, and we're not going to really look at families or orders or classes or phyla, but you should be able to put these in order. So be ready to put these in order and be familiar with them enough to do that. And you can look up mnemonic devices on the web for keeping kingdom, phylum, class, order, keeping those straight and in the right order. I recommend doing that. One thing you might enjoy looking through is the taxonomic levels and which group we are in within each of those levels. So, for example, you are a member of the species Homo sapiens in the genus Homo, and your family is Hominidae in the order Primates, in the class Mammalia, in the phylum Chordata, kingdom Animalia, domain Eukarya. Those are your groups for each of these taxonomic levels. So what I want to kind of illustrate with this table, though, is like what else is in those groups with us and how different are they? So this can give you some sense of what it means to be in the same domain or the same species. Within the species, there are no other species of, there are no other strains. There are no strains of Homo sapiens. So there's nothing else in our species but us. In the genus Homo, there are Homo sapiens like us, but there's nothing else currently living. But there are fossil, extinct other species within Homo. Homo neanderthalis, the Neanderthals, and Homo floresiensis. These are the hobbits. They were very small. They were on an island that got, or their habitat became an island, and so they ended up being something like 3 feet tall. But yeah, they were within our genus Homo. And then if you go all the way up to the domain level, within our domain, there are also plants like oak trees and molds like Aspergillus. So think about how different you are from an oak tree or a mold, but you are all in the same domain with them. And then if you go back down to something like order, Well, what's in that order Primates with us? Lemurs and monkeys. Those kind of look like us. And then if you go down to our family, which is a smaller group, that's where we find chimpanzees and orangutans, but not lemurs, not monkeys, only these bigger primates that look more like us. And if you go into our genus, and if you look at reconstructions of what Neanderthals and hobbits look like, they looked mostly like us. So the farther down this chart you go, the more each group is smaller and the members of the group are similar to each other. As you go up the group, they get bigger and bigger, and the members of the group might be really distant from each other. So what I want you to do with this is learn this trend, learn what it means to be a domain. It's such a huge group. Learn what it means to be a species or a genus. And this is an example of the same thing for E. coli 0157H7, for that strain. It just shows you for the different taxonomic levels, its group. Don't memorize this. You don't have to. And if you don't recognize the names of any of these microorganisms, don't worry about it. It's just there if you're curious. All right. Well, that's it for taxonomy. Taxonomy includes how we write organisms, but also the taxonomic levels. So again, the taxonomic levels you need to think about the most are domain, genus, species, and strain, but you need to be able to put all the taxonomic levels in order. If I were to give you an out of order list. All right. I'll see you in the next video.